ಮೇಡಮ್ ಶುರು ಮಾಡೋದಾ? ಹಲೋ ಹಲೋ ಶುರು ಮಾಡೋದಾ ಮ್ಯಾಮ್? ಹಲೋ ಹಲೋ
ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಇವಾಗ ಆಡಿಬಲ್.
Yes sir. Thank you.
ಶುರು ಮಾಡೋದಾ ಮ್ಯಾಮ್?
Yes sir, definitely.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಈ ಸಂಚಿಕೆ ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಶ್ರೀಮತಿ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿಯವರು ಕ್ಯಾನ್ಸರ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು ಅವರನ್ನು ಈ ವೇದಿಕೆಗೆ ಸ್ವಾಗತಿಸುತ್ತಾ ವೆಲ್ಕಮ್ ಟು ಮ್ಯಾಮ್.
Thank you so much sir, and good evening dear students. Thank you so much sir.
ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್. ಅದೇ ರೀತಿ ಇವತ್ತಿನ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಾಗಿರ್ತಕ್ಕಂಥ ನನ್ನ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರಿಗೂ ಕೂಡ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಾ ನಮ್ಮ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವರ ಬಗ್ಗೆ ಒಂದೆರಡು ಮಾತುಗಳು. ಸೊ Lakshmi Murthy ma'am completed his MSc Botany and also BA certificate in counseling skills, and now a senior biology teacher and trainer and adolescent counselor with over 25 years experience. Currently the program head of medical excel at Base Educational Services Private Limited and HOD of biology since 2018. In addition, senior biology faculty at Base Educational Service Private Limited since 2016.
Thank you sir. Thank you.
Some work recognition and received personal commendation letters from Ministry of HRD, Government of India for my contribution to education. Also, the best biology teacher award from the Indian Brain Bee organization. Also received the Reliance Foundation award for my contributions to biology teaching, and secured the first rank for senior school biology in the Teachers Professional Olympiad TPO for 2018 and 2019 at the All India of plus UAE level. May I have some educational history? BSc Chemistry, Botany Zoology from RLS Institution, Bangalore. MSc Botany, specialization in biotechnology and tissue culture from Karnataka University, Dharwad. And also have B.Ed in English and Science from ICFA. Certificate in Counseling Skills from Prasanna Counseling Center and NIMONS. And Development to Behavior from University of Maryland. Ma'am, I have some work experience, taught middle school chemistry and biology at Phoenix School, Belagaum. Also, biology for secondary school and senior school as a senior faculty at National Public School, Indiranagar and Rajajinagar from May 2000 to October 2015. And she was senior school student counselor at the National Public School, Rajajinagar, and the YET and senior coordinator and guide for the adolescent education program at the National Public School, Rajajinagar. She was the head and a senior coordinator for the drug abuse awareness program at the National Public School, Rajajinagar, and the head and senior coordinator for the continuous and comprehensive education program at the National Public Schools. Also, she was the teacher trainer for the CCE, AEP, and the life skills program at the National Public Schools. Mavs, hobbies, and interests. A trained dramatist and Carnatic vocalist. I enjoy reading and artwork. Also spend lecture time on writing short stories. Thank you, ma'am. Over to you.
Thank you so much, sir. Thank you so much. And welcome to the students. Today, we are going to be looking at a very important topic. And this topic is coming under a chapter which is called as Human Health and Disease, which is part of second year. Right? And this particular topic has got a lot of importance when it comes to the PU board exam, when it comes to Karnataka CET, when it comes also to the NEET exams. To a large extent, this topic is also tested in other international exams like the Olympiads. Okay, even in students who would want to go to pure science institutes, there will be certain entrance exams which are conducted by a number of colleges. Even in those colleges, you find that there will be few questions which come on this chapter, which is human health and disease, and particularly this disease, which is cancer, right? From that, even students of 11th standard will be interested to know about the disease cancer, because in first year biology, whether it is CBSE 11th standard or PUC, you find that there is a topic which is called as mitosis. And students will know that when mitosis goes out of control, it can lead to the disease called as cancer. So, with that brief introduction with respect to the importance of this disease cancer, let us immediately start off and try to learn as much as possible today on this disease, that is cancer. Right? Now, in order to understand what is cancer, it is important for students to understand the behavior with respect to the structure and division when it comes to normal cells. So let us see how normal cells behave. When you consider normal cells, right, these are normal living cells in the body. When you consider normal cells, you realize that normal cells have got normal structure and function. Normal cells will show normal structure and function. For example, consider a particular WBC, let us say, called as B lymphocyte, or consider a WBC called as T lymphocyte. what you notice is it has got a certain structure. For example, if I consider a B lymphocyte, it has got a certain structure. It is going to be a circular cell. Apart from that, there is going to be a very big nucleus that it has. It has got certain functions. For example, a B lymphocyte is going to manufacture the weapons for your body, which are called as antibodies, etc. So what you notice is a normal cell will show a normal structure and a normal function. That is the first thing that you notice in a normal cell. Apart from that, what you see is normal cells always show a controlled method of division. What does it mean? Controlled method of division. For example, all of you are aware that your skin gets shed frequently. Okay, so on a daily basis, cells from your body get shed off and your skin gets replaced frequently. The lining of the gut andre ಏನು right from the esophagus right up to the anus, we have a lining of the gut that also gets shed frequently, right? So these are certain tissues which show regular cell division. You can say almost on a daily basis the cells will divide. So what else we notice is in case of normal cells, they show normal and controlled method of division. There is a normal and controlled method of cell division that these normal cells are going to show, right? For example, you know that your skin cells will divide on a daily basis, whereas your bone cells will not divide on a daily basis. If there is a fracture, they will divide. Or if it is a young child whose bones are still to grow due to the growth hormone stimulation, then you find that the cells will be able to divide. So that means there is some kind of control which is going to be there when the cell should divide, how much it should divide, etc. Apart from that, normal cells are going to show a property which is called as contact inhibition. Let me explain what contact inhibition is going to be. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಒಂದು ಸಿಂಪಲ್ ಒಂದು ಕಂಡೀಷನ್ ನ ನೋಡೋಣ. ಓಕೆ, ಲೆಟ್ ಅಸ್ ಸೇ ಈಗ ನಂದು ಕೈಯಲ್ಲಿ, ಓಕೆ, ಇಫ್ ಯು ಕನ್ಸಿಡರ್ ಮೈ ಹ್ಯಾಂಡ್, ಲೆಟ್ ಅಸ್ ಸೇ ದೇರ್ ಇಸ್ ಅ ಸ್ಮಾಲ್ ಕಟ್ ಇನ್ ದ ಹ್ಯಾಂಡ್, ರೈಟ್? ಸೊ ಸ್ಮಾಲ್ ಕಟ್ ಇನ್ ದ ಹ್ಯಾಂಡ್ ಅಂದ್ರೆ ಏನು? ದಿಸ್ ಇಸ್ ಅ ಕಟ್ ವಿಚ್ ಹ್ಯಾಸ್ ಹ್ಯಾಪನ್ಡ್ ಇನ್ ದ ಹ್ಯಾಂಡ್ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಈಗ ಈ ಕಟ್ ಅಲ್ಲಿ ಲೆಟ್ ಅಸ್ ಸೇ 1 million cells have to be formed. It's a small number that I'm taking because it is just a fictitious condition. So if you consider this is a cut in the body, you let us say we have to make some 1 million cells to heal the injury. For wound healing, 1 million cells have to be produced, let us assume. ಈಗ ಈ 1 million cells produce ಮಾಡಕ್ಕೆ ಈ ಕಡೆಯಿಂದಾನೂ ಸೆಲ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ, ಈ ಕಡೆಯಿಂದಾನೂ ಸೆಲ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ. ಆ ಸೆಲ್ಸ್ ಒಂದಕ್ಕೊಂದು ಕಾಂಟ್ಯಾಕ್ಟ್ ಮಾಡಿದ ತಕ್ಷಣ ಏನಾಗುತ್ತೆ? ಇಮಿಡಿಯೇಟ್ಲಿ ದೇ ವಿಲ್ ಸೇ, oh, cells are being produced, let us now stop dividing. So when cells come in contact with one another, immediately they will realize that it is time to stop dividing because the number of cells have already been produced as much as was required. That type of inhibition of cell division due to contact with each other is called as contact inhibition. ನಿಮ್ಗೆ ಅರ್ಥ ಮಾಡಿಸ್ಬೇಕಾದ್ರೆ ಒಂದು ಗೋಡೆ ಕಟ್ಟಬೇಕಾಗಿದೆ. ಇಬ್ರು ಜನ ಗೋಡೆ ಕಟ್ತಾ ಇದ್ದಾರೆ. ಈ ಕಡೆಯಿಂದ ಒಬ್ಬ ಮನುಷ್ಯ ಬ್ರಿಕ್ಸ್ ಇಡ್ತಾ ಇದ್ದಾನೆ, ಈ ಕಡೆಯಿಂದ ಒಬ್ಬ ಮನುಷ್ಯ ಬ್ರಿಕ್ಸ್ ಇಡ್ತಾ ಇದ್ದಾನೆ. ಅದ್ ಹತ್ರ ಬಂದ ತಕ್ಷಣ ಅವರು ಗೋಡೆ ಕಟ್ಟೋದು ನಿಲ್ಲಿಸ್ಬಿಡ್ತಾರೆ. ಯಾಕಂದ್ರೆ ಬ್ರಿಕ್ಸ್ ಹಾವ್ ಕಮ್ ಇನ್ ಕಾಂಟ್ಯಾಕ್ಟ್ ವಿತ್ ಒನ್ ಅನದರ್. ಅದೇ ತರಹ normal cells, when they divide, when they come in contact with one another, they will stop dividing. That is called as contact inhibition. And normal cells have these functions and structure and this property called as contact inhibition, and they are able to do that because their cell division is going to be controlled. Next question immediately barodu, who who is going to control cell division? Immediately the question is going to come. So cell division is going to be controlled by certain chemicals which are called as mitogens. Cell division, who is going to control? Cell division in normal cells, cell division is going to be controlled by certain chemicals called mitogens. These are the chemicals. In the presence of these chemicals, mitosis will occur. If these chemicals are not there, you will realize that mitosis will not occur. So you find that cell division is under the control of chemicals called mitogens. E particular chemical ಹೆಸರು you may not find in NCERT book, but it is important to understand that there are certain chemicals which can control cell division. Next question that arises is, who is going to control the release of mitogens? Which, is there a gene present? Who is going to control these mitogens? Definitely what you realize is these chemicals, their production, their breakdown, everything is going to be controlled by genes. And the genes which control normal cell division in normal cells are of two types. What are the two types of genes that control normal cell division? The genes are, first of all, you have proto-oncogenes. Proto-oncogenes are going to be the genes which control normal cell division. So proto-oncogenes will control normal cell division. So they are the ones which will make the cell go into the stages of cell division, right? Apart from that, the second group of cells which control normal cell division are going to be called as TSG. What is this TSG? TSG is nothing but tumor suppressor genes. Tumor suppressor genes. These TSG also control cell division. How do they control? Proto-oncogenes are going to control actual cell division. Tumor suppressor genes are going to control at the rate of which cell division will occur and how many cells are going to be produced. ಮತ್ತೆ ಹೇಳ್ತೀನಿ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Proto-oncogenes will control cell division steps. Tumor suppressor genes will control at what rate the cell has to divide, as well as how many cells should be produced under cell division. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನೀವು ನೋಡಿರ್ಬೋದು, ಈಗ ಲೆಟಸೆ ನಂಗೆ ಒಂದು ಚಿಕ್ಕ ಕಟ್ಟಾಯ್ತು ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ಬೆಟ್ ಮೇಲೆ ಪುಟ್ಟ ಕಟ್ಟಾಯ್ತು ಚಾಕುವಿಂದ. ಯು ವಿಲ್ ರಿಯಲೈಸ್ ದಟ್ ಇನ್ another 4-5 hours that cut is healed. ಓಕೆ, ಚಿಕ್ಕ ಕಟ್ಟಾಗಿತ್ತು, 4-5 hours ಅಲ್ಲಿ ಆ ಕಟ್ ಹೀಲ್ ಆಗೋಯ್ತು. ಅಷ್ಟೇ ಸ್ಮಾಲ್ ಕಟ್ ಸಪೋಸ್ ನನಗೆ ನನ್ನ ಏಜ್ ಅಲ್ಲಿ ನನ್ನ ಬೋನ್ ಗಾಯ್ತು ಅಂದ್ರೆ ಇಟ್ ವಿಲ್ ಟೇಕ್ ಅಟ್ ಲೀಸ್ಟ್ 4 to 5 weeks ಫಾರ್ ದ ಕಟ್ ಟು ಹೀಲ್. ಅದಕ್ಕೆ ಫ್ರಾಕ್ಚರ್ ಅಂತೀವಿ, ಕರೆಕ್ಟಾ? ಅದು ಹೀಲ್ ಆಗ್ಬೇಕು, ಹೊಸ ಸೆಲ್ಸ್ ಆ ಗ್ಯಾಪ್ ಅಲ್ಲಿ ಫಿಲ್ ಆಗ್ಬೇಕು ಅಂದ್ರೆ ಇಟ್ ವಿಲ್ ಟೇಕ್ 4 to 5 weeks. ಹೂ ಇಸ್ ಕಂಟ್ರೋಲಿಂಗ್ ದಿಸ್ ರೇಟ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್? That is tumor suppressor genes. Understand? So, ಒಂದು analogy ಕೊಡಬೇಕಾದ್ರೆ, ನಿಮ್ಮ proto-oncogenes are like the accelerator. They want to see that cell division should occur. But ಹೌದು, ಕಾರ್ ಮುಂದೆ ಹೋಗ್ಬೇಕು, ಬಟ್ ಇನ್ ಅ ಕಂಟ್ರೋಲ್ಡ್ ಮ್ಯಾನರ್ ಇಟ್ ಶುಡ್ ಗೋ ಫಾರ್ವರ್ಡ್, ಕರೆಕ್ಟ್? ಸೊ ಅದನ್ನ ಯಾರು ಕಂಟ್ರೋಲ್ ಮಾಡ್ತಾರೆ? ದಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ದ ಬ್ರೇಕ್. ಸೊ ಟ್ಯೂಮರ್ ಸಪ್ರೆಸರ್ ಜೀನ್ಸ್ ವಿಲ್ the formation of a tumor or extra cells. So idu brake thara, it is keeping a control on how the car is going to move. Is that clear? So this is going to be normal cells. Ega let us see cancer agbekadre what is going to happen. Let us try and understand. Okay, now this is how it is going to work. For a cell to turn cancerous, for a cell to turn cancerous, what exactly is going to happen? Let us try and understand. This is how it is going to work, right? So firstly, you must have, I have spoken about these genes, right? So there are these genes present. These genes are going to turn into mutated genes. Mutation andre ಏನು? It is like a spelling error in a gene. It is like a change in the gene. So genes are going to become mutated genes. ಯಾವ genes ಅಂತ ನಾ explain ಮಾಡಿದ್ದೀನಿ. That is tumor suppressor genes as well as your proto-oncogenes. So what is going to happen? Genes will become mutated genes when they are exposed to certain chemicals called as carcinogens. When certain agents called as, sorry, chemicals better, agents. So genes get converted to mutated genes due to the exposure to some agents called as carcinogens. What are these carcinogens? So carcinogens, as I said, are going to be the generators of cancer. So they are the ones which are going to bring about cancer formation, right? That's why they are called generators of cancer. Carcin andre ಏನು ಅಂತ ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಟೈಮಲ್ಲಿ ಹೇಳ್ತೀನಿ, right? What are the usual carcinogens, or which agents can bring about cancer? Let us try and see. So the agents that can bring about cancer can be in the form of radiations. So some radiations can cause cancer. Which radiations can cause cancer? For example, X-rays can cause cancer, gamma rays can cause cancer, UV rays can cause cancer, etc. So there are certain radiations which can cause cancer. That means exposure to these radiations can convert the genes and make them mutated. What is the name of the mutated genes? I'll be able to tell you. Okay, next is going to be, there may be certain chemicals also. Okay, ನೀವು ನೋಡಿರಬಹುದು, if you want to go and buy a soap ಇಲ್ಲ ಒಂದು ಶಾಂಪೂ ಕೊಂಡ್ಕೋಬೇಕಾದ್ರೆ ಅದ್ರ ಮೇಲೆ ಏನ್ ಬರ್ದಿರುತ್ತೆ? It is paraben free ಅಂತ ಬರ್ದಿರುತ್ತೆ. Parabens have been found with research to be cancer causing agents. ಅದೇ ತರ there are other kinds of chemicals. For example, ನೀವು ನಿಕೋಟಿನ್ ಬಗ್ಗೆ ಕೇಳಿರಬಹುದು, or cigarettes ಅಲ್ಲಿ there are many other types of hydrocarbons which can cause cancers. ಇಲ್ಲ ಅಂದ್ರೆ second year students would have heard about a particular chemical called as ethidium bromide. Biotechnology chapter al barutte. Ethidium bromide is a highly cancer causing agent, right? So athara there may be certain chemicals which are considered to be capable of converting their genes into mutated genes. Ade thara there are some viruses which are also considered to have carcinogenic effect. ನಾವು ಹೆದರ್ಕೋಬೇಕಾಗಿಲ್ಲ, ಓ viruses ಇಂದ ಕ್ಯಾನ್ಸರ್ ಬರುತ್ತೆ ಅಂದ್ರೆ ಹಂಗಾರೆ is cancer communicable? It's not like that. There are some viruses which are capable of introducing their genes which can help in unlimited cell division. Avu papa, their intention was not to cause cancer. The viral intention was just to make more host cells. But in the process of that, sometimes it may lead to cancer. So there are some viruses. For example, there is a virus called as HPV, human papilloma virus anta heltivi. So this is implicated in genital warts as well as cervical cancer. Okay, so like that, there may be certain viruses also present which can actually, cause cancer. Okay, now we have to understand exposure to these are going to convert your proto-oncogenes into what genes? So the mutated genes, normal genes are proto-oncogenes, mutated genes are called as C-onc. C-onc andre cellular oncogenes. Similarly, viruses will contain their own genes called as V-onc genes or viral oncogenes, which can dominate over the proto-oncogenes. As a result of which, the dominated proto-oncogene, which has got converted to C-onc, or you find the V-onc gene, is going to not only bring about unlimited cell division, it will also suppress the tumor suppressor genes, as a result of which tumor suppressor genes it andre accelerator brake ne fail madbidtu. ಆ ಥರ the C-onc genes or the V-onc genes can not only bring about unlimited cell division, they will also suppress the tumor suppressor genes so that there is no control on the rate of cell division, there is no control on the number of cells produced. So this is how exposure to these chemicals is going to be the cause for chemicals or radiations or viruses is going to be the cause for cancer. ಓಕೆ ಇಮಿಡಿಯೇಟ್ ಕ್ವೆಶ್ಚನ್ ಬರುತ್ತೆ ಇವಾಗ ಹೊರಗಡೆ ಹೋದ್ರೆ ಯು ವಿ ರೇಸ್ ಇದೆ ಎಲ್ಲಾರ್ಗೂ ಯಾಕೆ ಕ್ಯಾನ್ಸರ್ ಆಗಲ್ಲ ಇಲ್ಲ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಸೋ ಮೆನಿ ಪೀಪಲ್ ಆರ್ ಸ್ಮೋಕಿಂಗ್ ಸಿಗರೆಟ್ಸ್ ವಿಲ್ ಎವ್ರಿಬಡಿ ಗೆಟ್ ಕ್ಯಾನ್ಸರ್ ಸೊ ಈ ಕ್ವೆಶ್ಚನ್ ಬಂದೇ ಬರುತ್ತೆ ಪ್ಲೀಸ್ ಟ್ರೈ ಅಂಡ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ what is basically happening is there is a doubt bold you're not able to see the board are you seeing in a phone or something sir if you can make my screen bigger and ತಮ್ಮ ಸ್ಕ್ರೀನ್ ನ ಚಿಕ್ಕದ್ ಮಾಡ್ಬಿಟ್ರೆ ದೆನ್ ಮಕ್ಕಳು ಕ್ಲಿಯರ್ ಆಗಿ ನೋಡ್ಬೋದು ಅನ್ಸುತ್ತೆ ಒಂದ್ಸತಿ ಚೆಕ್ ಮಾಡಿ ಆ ಪಾಸಿಬಿಲಿಟಿ ಇದೆಯಾ ಅಂತ ಐ ಡೋಂಟ್ ನೋ ವೆದರ್ ಐ ಹ್ಯಾವ್ ದ ಕಂಟ್ರೋಲ್ ಆಫ್ ಮೇಕಿಂಗ್ ಮೈ ಸ್ಕ್ರೀನ್
ನೀವು ಫುಲ್ ಸ್ಕ್ರೀನ್ ಮಾಡಿದ್ರೆ ಮ್ಯಾಮ್
ಅದನ್ನ ಹೇಗೆ ಮಾಡೋದು ಅಂತ ನಂಗೆ ಇದು ಇದ್ರಲ್ಲಿ ಮೂವ್ ಟು ಸ್ಟೇಜ್ ಅಂದ್ರೆ ಆಗತ್ತಾ ಇಲ್ಲ ಸ್ಟೇಜ್ ಆಗಲ್ಲ
ಸ್ಟೇಜ್ ಮಾಡಿದ್ದೀನಿ ನೀವು ಕೊಡಿ ಅಲ್ಲಿ ಮ್ಯಾಮ್
ಮಾಡಿದ್ದೀನಿ ಮೂವ್ ಟು ಸ್ಟೇಜ್ ಮಾಡಿದ್ದೀನಿ ನಾನು
1 ಮಿನಿಟ್ ನೀವು ಕಂಟಿನ್ಯೂ ಮಾಡಿ ಮ್ಯಾಮ್ ನಾನು ಒಂದು 2 ನಿಮಿಷ ಮಾಡಿಸ್ತೀನಿ
yes right so as a result you find that ಮಕ್ಕಳೇ ನೀವು ಯೋಚನೆ ಮಾಡ್ಬಿಡಿ ನಾನು ಹೇಳ್ತಾ ಇರೋ ಇನ್ಫಾರ್ಮೇಶನ್ ಎಲ್ಲ ನಿಮ್ಮ NCERT ಬುಕ್ ಅಲ್ಲಿ ಇದೆ ಓಕೆ ನಾನು ಏನ್ ಹೇಳ್ತಾ ಇದ್ದೀನೋ ಅದು ನಿಮ್ಮ NCERT ಬುಕ್ ಅಲ್ಲಿ ಇದೆ yes Yogesh has asked very good question Yogesh can all radiations cause cancer illa Yogesh iga think about radio waves ಬಗ್ಗೆ ಯೋಚಿಸು so all radiations cannot cause cancer illa namma visible light rays ಬಗ್ಗೆ ಯೋಚಿಸು they all will not cause cancer artha aita okay now going ahead we are so many people are going to be exposed to these chemicals etc so if that is the case what we need to know is how come everybody does not get cancer okay ಒಂದು ನಾವು ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕು children it is like this what you realize is ಪ್ರತಿಯೊಬ್ಬರಲ್ಲೂ ಬೇರೆ ಬೇರೆ ಜೆನೆಟಿಕ್ ಮೇಕಪ್ ಇರುತ್ತೆ ಸೊ some people have got excellent immunity because as soon as one cell turns cancerous immediately you will realize that the WBC will be able to identify this cancerous cell because ನಾನು ಹೇಳ್ದಂಗೆ cancerous cell structure ಬೇರೆ ಇರುತ್ತೆ I'll come to that so they will be able to identify ಮಾಡಿ they can actually destroy the cancer cell so it is very easy to actually destroy a cancer cell WBC ne destroy ಮಾಡ್ಬಿಡುತ್ತೆ okay and apart from that some people have got good lifestyle also not just good genes they have good lifestyle ಯಾವ ತರ ಅಂದ್ರೆ ಒಳ್ಳೆ fruits and vegetables ತಿಂತಾ ಇರ್ತಾರೆ ಹಾಗಾಗಿ ಒಳ್ಳೆ antioxidants ಇರುತ್ತೆ once there are good antioxidants you find that it becomes very easy not to have this kind of mutation take place right so ee thara small changes in lifestyle will also help to postpone cancer as much as possible viruses cause mado cancer na for example iga HPV ge vaccine banbitide cervical cancer vaccine anta ಹೇಳ್ತೀವಿ yes sir ಹೇಳಿ
it's full screen
ಆಗಿದೆಯಾ okay done thank you children are able to see andre agutte nange I will not be able to see nandu hengideyo hange irutte children are able yes okay thank you so much so makle as I said all people who get exposed to these carcinogens need not get cancer but nobody will take the risk of course ad nimgu gottide But what we have to understand is the genotype of the person, ನಮ್ಮ genetic makeup, and apart from that our lifestyle. ನಾನು ಹೇಳ್ದಂಗೆ ಒಳ್ಳೆ ಫ್ರೂಟ್ಸ್ ಅಂಡ್ ವೆಜಿಟಬಲ್ಸ್ ತಿನ್ನೋದಿರ್ಬೋದು, ಜಂಕ್ ಫುಡ್ ಆಸ್ ಮಚ್ as ಪಾಸಿಬಲ್ ಅವಾಯ್ಡ್ ಮಾಡೋದಿರ್ಬೋದು, ಎಕ್ಸ್ಪೋಜರ್ ಟು ಸಿಗರೆಟ್ ಸ್ಮೋಕ್ ಇರ್ಬೋದು, ಇಲ್ಲ ಅಂದ್ರೆ ಈಗ ಹೊರಗಡೆ ಹೋದಾಗ ಸನ್ಸ್ಕ್ರೀನ್ ಹಾಕೋತಾರೆ ಒಬ್ರೊಬ್ರು ಜನ. ಓಕೆ, ಸೊ ಈ ತರದ್ದು ಬೇರೆ ಬೇರೆ ಸ್ಮಾಲ್ practices will help to see that cancer can be kept off. But apart from that, how far the mutation has gone anta irodu. Iga brake fluid du wire cut madbitre, whatever you do, you find that it is not going to be easy for the for cancer to be controlled. It is going to go out of control completely, right? Let us now go ahead and see what is going to be the features of cancerous cells. What is going to be the features of cancerous cells? So cancerous cells, these are the features that cancerous cells have. Firstly, nimge gottirbodu, they do not have contact inhibition. They do not have contact inhibition. Andre enu, when they come in contact with one another, they will continue to divide. When they come in contact with one another, they will continue to divide. They cannot stop dividing. So they will not have contact inhibition. Because there is no contact inhibition, you will find there is going to be no control over how many cells are produced. As a result of this, there will be excessive cells produced, and those excessive cells will be called as a tumor. They are called as a tumor. Because there is no contact inhibition, excessive cells are produced, and these excessive cells will produce, you can say, a mass which is going to be called as a tumor. So that is the first property of cancerous cells. Second property of cancerous cells is they do not have normal structure and function. Normal structure as well as function is not going to be seen in cancerous cells. What does it mean normal structure and function is not there? For example, some of the students may have heard about a disease called leukemia, blood cancer anta heltivi. Leukemia li the person is going to have a lot of WBC, much more than what is expected to be a normal value of WBC. Tumba WBC agbidutte. What happens when there is a lot of WBC? Somebody may say, oh, WBC is meant for defense. ಜಾಸ್ತಿ WBC ಆದ್ರೆ ಒಳ್ಳೇದಲ್ವಾ, immunity better ಆಗುತ್ತೆ ಅಂತ. No, it is wrong. These WBC may be more in number, but they are not able to do their work because they do not have normal function. They don't have normal structure also, and they do not have normal function also. So that becomes a big problem. You understand that? So this is another property of cancerous cells. Next property of cancerous cells is they are going to have very high rate of cell division. They will have extremely high rate of cell division. So cell division rate is going to be extremely high. So uncontrolled cell division is going to occur. So not only do they stop dividing, they will not do their function also properly, and they just continue to divide more and more and more. This is the property of cancerous cells. Now, based on tumor ಎಷ್ಟು ದೊಡ್ಡದಾಗಿದೆ, ಎಷ್ಟು ಫಾಸ್ಟ್ ಆಗಿ ಗ್ರೋ ಆಗ್ತಾ ಇದೆ ಅನ್ನೋ ಪ್ರಕಾರ we can classify tumors. I'm going to go to classification of tumors. We can classify tumors into two categories. Based on what, children? Based on what is the speed or rate of cell division, and also on the on whether that particular tumor is local or whether it can spread. I'll repeat again. So based on rate of cell division, ನಾನು ಡಿಕ್ಟೇಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸೊ ಹಂಗಾಗಿ ದೋಸ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಹೂ ಆರ್ ನಾಟ್ ಏಬಲ್ ಟು ಸಿ ದ ಬೋರ್ಡ್, ಮೇ ಬಿ ನಿಮ್ಮ ಹತ್ರ ಚಿಕ್ಕ ಫೋನ್ ಇದೆ, ಕಾಣಿಸ್ತಾ ಇಲ್ಲ ಅಂದ್ರೆ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಹಿಯರ್ ಮಿ. ಓಕೆ, ಸೊ ಬೇಸ್ಡ್ ಆನ್ ದ ರೇಟ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್ ಆಸ್ ವೆಲ್ ಆಸ್ based on the location of the tumor, you find that tumors are going to be of two types. What are the two types of tumors? The first type of tumor is called as benign tumor. The first type of tumor is called as benign tumor. What exactly is a benign tumor? Benign tumor, rate of cell division is a little less. ತುಂಬಾ ಫಾಸ್ಟ್ ಆಗಿ ಗ್ರೋ ಆಗಲ್ಲ. Second feature of benign tumor is going to be that it is localized. It is present only in a particular location. It will not spread to other parts of the body. ಮತ್ತೆ repeat ಮಾಡ್ತೀನಿ. Benign tumor means the tumor has got a slightly lower rate of cell division. Apart from that, this tumor is going to be localized. So it is restricted to a certain part of the body. It will not spread. That is called as a benign tumor. Iga, you must have heard about, example is, you must have heard about moles. Moles, okay? ಒಬ್ರೊಬ್ರಿಗೆ ಈ ಸಂಗಾತಿ ತರ ಇರುತ್ತೆ. Okay, that is called as a mole. That is going to be a benign tumor. ಅದೇ ತರ ಬೇರೆ benign tumors ಇದೆ. ತಾವು MBBS ಎಲ್ಲ ಓದಿದಾಗ you will be able to learn it in detail. So one of the benign tumors is going to be called as a mole. Right? So here you've noticed moles irutte, lipoma irutte, ಈ ತರ ತುಂಬಾ benign tumors ಬರುತ್ತೆ. So here the growth is little less and it is localized. ಬೇರೆ ಕಡೆ spread ಆಗಲ್ಲ. That is a benign tumor. Benign tumor ಇಂದ usually harm ಆಗಲ್ಲ. Okay? Benign tumor ಇದ್ದೋರ್ಗೆ ನಾವು ಕ್ಯಾನ್ಸರ್ ಇದೆ ಅಂತ ಹೇಳಲ್ಲ. We just say that they have a mole. ಅಷ್ಟೇ ಹೇಳ್ತೀವಿ. ಬಟ್ ಕ್ಯಾನ್ಸರ್ ಅನ್ನೋ ವರ್ಡು ಯೂಸ್ ಮಾಡಲ್ಲ. ಇಟ್ ಇಸ್ ನಾಟ್ ಯೂಸ್ ಬಿಕಾಸ್ ಇಟ್ಸ್ ನಾಟ್ ಅ ಡಿಸೀಸ್. ಬಿನೈನ್ ಟ್ಯೂಮರ್ಸ್ ಯೂಶಲಿ ಆರ್ ನಾಟ್ ಹಾರ್ಮ್ಫುಲ್. ವೆನ್ ಕ್ಯಾನ್ ದೇ ಬಿ ಹಾರ್ಮ್ಫುಲ್? ಡಿಪೆಂಡಿಂಗ್ ಆನ್ ದ ಲೊಕೇಶನ್. ಅಂದ್ರೆ ಏನು? ಈಗ ಬ್ರೈನ್ ಒಳಗಡೆ ಒಂದು ಬಿನೈನ್ ಟ್ಯೂಮರ್ ಇದೆ ಅಂತಂದ್ರೆ ಇಟ್ ಮೇ ಬಿ ಪ್ರೆಸ್ಸಿಂಗ್ ಆನ್ ಅನ್ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾರ್ಟ್, ನಾಟ್ ಅಲವಿಂಗ್ ದಟ್ ಪಾರ್ಟ್ ಟು ಫಂಕ್ಷನ್. So depending on such critical locations, sometimes benign tumors also may be dangerous, right? Next type of tumor is called as malignant tumor. What is malignant tumor? Malignant tumor is the one where the rate of cell division is going to be very high. Very fast the cells are going to divide. That is first thing. Second thing, what you notice in malignant tumor is very simple. For example, let us say there is a tumor which is present in a particular location. These are all the tumorous cells which are present in a particular location. And next to that, there is going to be a lymph vessel or a blood vessel, let us say. What is going to happen? These cells will become loose. They will enter into this blood vessel and they will migrate to another place and set up a new tumor there. ಮತ್ತೆ ಹೇಳ್ತೀನಿ. So in malignant tumor, you find that the rate of cell division is high. Apart from that, what you notice is the cells can enter into the lymph system or the blood system and they will migrate via the blood or lymph to another part of the body, and in the other part of the body they will set up a new tumor. This particular property where the cancer can spread to other parts of the body is called as metastasis. This property is called metastasis, where you notice that the cancer cells have migrated to other parts of the body and they have set up a new tumor there. ಇದಕ್ಕೆ metastasis ಅಂತ ಹೇಳ್ತೀವಿ. Okay, and that happens to be one of the most, you can say, feared properties or dreaded properties of the disease cancer, because the doctors will not know where exactly to treat. So they will have to increase the treatment. They will have to make sure that many parts of the body get treated. Okay, I'm not saying that treatment is not possible. India, due to efforts of a number of highly respected doctors, cancer treatment detection has become so good, no doubt about it. But still what you realize is cancer is taking many lives all over the world every year. It is one of the leading causes of non-infectious death in the world, and that is mainly because of this metastasis. And that is how they are able to move to other parts and they are able to settle down. ಏನಾಗುತ್ತೆ when this tumor is growing, ಈಗ ನಮ್ಗೆ ಅನ್ನಿಸ್ಬೋದು, ಈಗ think about somebody who has got a sixth finger. ಒಂದು some million cells are extra. How is that going to harm the body? ಅದೇ ತರ ಈಗ ಯಾರಿಗೋ ಕಿಡ್ನಿ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟ್ ಆದಾಗ ಅವರಲ್ಲಿ 3 ಕಿಡ್ನಿ ಇರುತ್ತೆ. ಓಕೆ, 3 ಕಿಡ್ನಿ ಇರುತ್ತೆ ಒಳಗಡೆ, ಒಂದು ಕೆಟ್ಟೋಗಿರೋದು, ಇನ್ನೊಂದು ಚೆನ್ನಾಗಿರೋದು, ಈ ತರ 3 ಕಿಡ್ನಿ ಇರೋ ಪಾಸಿಬಿಲಿಟಿ ಇದೆ. ಒಂಚೂರು ಎಕ್ಸ್ಟ್ರಾ ಕಿಡ್ನಿ ಬಂದ ತಕ್ಷಣ ಹೌ ಕ್ಯಾನ್ ಇಟ್ ಬಿ ಸೋ ಮಚ್? ಎಕ್ಸ್ಟ್ರಾ ಸೆಲ್ಸ್ ಇದ್ರೆ ವಾಟ್ ಇಸ್ ದ ಬಿಗ್ ಡೀಲ್? ದಟ್ ಇಸ್ ನಾಟ್ ಹೌ ಇಟ್ ಇಸ್. ದೀಸ್ ಕ್ಯಾನ್ಸರಸ್ ಸೆಲ್ಸ್ ಆರ್ ವೆರಿ ಹಂಗ್ರಿ ಫಾರ್ ನ್ಯೂಟ್ರಿಷನ್. So they will take up a lot of nutrition from the body. Because of that, normal cells start dying. And plus their rate of production is going to be at the cost of normal cells. Because of all these reasons, you find that cancer can actually kill, right? Okay, next question barodu. Okay, this is how I hope you understood what exactly will be the the features of cancerous cells. They have high rate of cell division. Apart from that, they also do not have normal structure and function. That's how your WBC will be able to identify them and destroy them. If it is possible, if the rate of division is too high, then WBC will not be able to control it. And I also told you about how contact inhibition is lost and adake tumor anta agutte. Okay, I was able to explain that. Okay, next question is going to be as to how to detect cancer. ಕ್ಯಾನ್ಸರ್ನ ನಾವು ಹೇಗೆ ಡಿಟೆಕ್ಟ್ ಮಾಡ್ಬೋದು? ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಪರ್ಸನ್, he has cancer, she has cancer ಅಂದ್ರೆ how to detect. So let us go to detection of cancer, or we can say diagnosis or detection of cancer. How can we do? That is going to be the next question. Okay, let us try and understand how to detect cancer. So I will explain one by one. ಯಾವ ತರ ಡಿಟೆಕ್ಟ್ ಮಾಡಬಹುದು ಅಂತ. ಓಕೆ, ಸೊ ದ ಫಸ್ಟ್ ಡಿಟೆಕ್ಷನ್ ಆಫ್ ಕ್ಯಾನ್ಸರ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಥ್ರೂ ಹಿಸ್ಟೋಪ್ಯಾಥಾಲಜಿ. ಇವತ್ತು ಬೈ ದ ಟೈಮ್ ವಿ ಕಮ್ ಟು ಅರೌಂಡ್ 4:45 ಬಂದ ತಕ್ಷಣ ನಾನು ನಿಮ್ಗೆ ಪವರ್ ಪಾಯಿಂಟ್ ಪ್ರೆಸೆಂಟೇಶನ್ ಪ್ರಿಪೇರ್ ಮಾಡಿದ್ದೀನಿ. ಓಕೆ, ನಾನು ನಿಮ್ಗೆಲ್ಲ ಇದರದ್ದು ಪಿಕ್ಚರ್ಸ್ನು ತೋರಿಸ್ತೀನಿ. ಓಕೆ, ರೈಟ್? ಸೊ ಹಿಸ್ಟೋಪ್ಯಾಥಾಲಜಿ is the first way to detect cancer. What is this histopathology? Let us try and understand. So let us say there is a person, for some reason or the other, he is not having good health. So tumba tiredness barta ide, lot of other types of symptoms are there. He is not able to eat properly. Tumba tharadu symptoms ide anta anಕೊಳ್ಳಿ. So doctor first ಏನ್ ಅನ್ಕೋತಾರೆ, they will feel liver is weak anta ಹೇಳ್ತಾರೆ. And they will start treatment. Liver is weak ಅಂತ ಟ್ರೀಟ್ಮೆಂಟ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರು ಏನು ಉಪಯೋಗ ಆಗ್ತಾ ಇರಲ್ಲ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಆವಾಗ ದೆನ್ ದ ಡಾಕ್ಟರ್ ಮೈಟ್ ಬಿ ಸಸ್ಪೆಕ್ಟಿಂಗ್, ಇವ್ರಿಗೆ ಲಿವರ್ ಕ್ಯಾನ್ಸರ್ ಇರಬಹುದು ಅಂತ ಅನ್ಕೋತಾರೆ. Then they will go for histopathology. How to do histopathology? Very simple. ನಾನು ಬೋರ್ಡ್ ಮೇಲೆ ಬರೆಯಲ್ಲ, ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ, ತಾವು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. So what is going to be done? ಒಂದು ಲಾಂಗ್ ಸಿರಿಂಜ್ ನ ತಗೊಂಡು they will insert it so that it goes into the liver. ಲಿವರ್ ಒಳಗಡೆವರೆಗೆ ಸ್ಕ್ಯಾನ್ ಮಾಡ್ತಾ ನೋಡ್ತಾರೆ, ಸುಮ್ನೆ ಹಾಕಕ್ಕಾಗಲ್ಲ. Through scans they will see where the liver is and they will insert the syringe and take out a small amount of liver tissue. ಲಿವರ್ ಟಿಶ್ಯೂನ ಆಚೆ ತಗೊಂಡ್ಮೇಲೆ that liver tissue, taking it out of the body, is called as biopsy. Taking a liver tissue out of the body. Liver tissue irbodu, or it may be somebody may be having too much of blood when they cough, so somebody might be suspecting there may be lung cancer anಕೊಳ್ಳಿ. So in that case, from the lung a small amount of the tissue can be taken. So wherever the cancer is suspected, small amount of the tissue is removed out. We call that as biopsy. What do we do? ಈ ತರ ಆಚೆ ತೆಗೆದ್ಮೇಲೆ ಏನ್ ಮಾಡ್ಬೇಕು? Very simple it is going to be. So that small tissue, let us say ಒಂದು ಚಿಕ್ಕ ಟಿಶ್ಯೂ ಹಿಂಗೆ ಆಚೆ ತೆಕ್ಕೊಂಡಿದ್ವಿ. That tissue, what do we do? ಅದನ್ನ we will put it into paraffin wax. ನೀವು ಕ್ಯಾಂಡಲ್ಸ್ ನೋಡಿದಿರಲ್ಲ, ಪ್ಯಾರಫಿನ್ ಕ್ಯಾಂಡಲ್ಸ್, ಆ ತರ ವ್ಯಾಕ್ಸ್ ಸಿಗುತ್ತೆ, ಹೈ ಕ್ವಾಲಿಟಿ ವ್ಯಾಕ್ಸ್. This tissue is going to be put inside paraffin wax. Once the tissue is put into paraffin wax, then they will make slices of it. ಓಕೆ, ಒಂದು ಮಷೀನ್ ಇದೆ ಮೈಕ್ರೋಟೋಮ್ ಅಂತ. ದೇ ವಿಲ್ ಮೇಕ್ ಸ್ಲೈಸಸ್ ಆಫ್ ಇಟ್. ಸ್ಲೈಸ್ ಮಾಡಿದ್ಮೇಲೆ ಪ್ಯಾರಾಫಿನ್ ವ್ಯಾಕ್ಸ್ ನೆಲ್ಲ ರಿಮೂವ್ ಮಾಡಿಬಿಟ್ಟು ದೇ ವಿಲ್ ಪುಟ್ ಸರ್ಟನ್ ಸ್ಟೇನ್ಸ್. ಸೊ ಸ್ಟೇನ್ಸ್ ಹಾಕಿದ್ರೆ ಏನಾಗುತ್ತೆ, ಇಮಿಡಿಯೇಟ್ಲಿ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಸಿ ದ ಸೆಲ್ಸ್. ರೈಟ್, ರಿಮೆಂಬರ್ ಡಯಾಗ್ನೋಸಿಸ್ ಲ್ಯಾಬ್ ಅಲ್ಲಿ ಇರ್ತಾರಲ್ಲ ಟೆಕ್ನಿಷಿಯನ್ಸ್, ದೇ ವಿಲ್ ನೋ ಹೌ ನಾರ್ಮಲ್ ಲಿವರ್ ಸೆಲ್ಸ್ ಲುಕ್ ಲೈಕ್, how normal lung cells look like. ಅದು ಅವ್ರಿಗೆ ಗೊತ್ತಿರುತ್ತೆ. Now they will see, oh, the cells look very different. The nucleus looks different, the shape looks different, the size looks different. They will be able to make out that definitely the cells are looking different. Adu nod takshana they will be able to identify that the cells are cancerous. Idakke ನಾವು histopathology anta antivi. Biopsy ಮಾಡಿ to study the cell under the microscope, and you have to compare it with normal cells and you will get to know whether it is cancerous or not. That is one method by which cancer can be diagnosed. Second method, idralli histopathology ಮಾಡಕ್ಕೆ, of course the students, ನೀವೆಲ್ಲ intelligent ಮಕ್ಕಳು, you know by now, for example, deep inside the brain if there is a tumor, and we have also seen it, okay, notice ಮಾಡಿದ್ವಿ ಅಂದ್ರೆ we will not be able to do histopathology. ಯಾಕಂದ್ರೆ ಏನಾಗ್ಬಿಡುತ್ತೆ, we cannot penetrate that part and we cannot remove a tissue. So sometimes it might be difficult to do biopsy also. Understand that. ಯಾವ್ದಾದ್ರು critical area damage ಆಗ್ಬೋದು. So ಹಂಗಾಗಿ histopathology ಎಲ್ಲಾ ಕಡೆನೂ ಮಾಡಕ್ಕಾಗಲ್ಲ. Depending on the cancer and the location, we will be able to do. Okay, suspected tumor ಎಲ್ಲಿದೆ, based on that we will be able to do. Okay, next is going to be called as a blood smear. There is one particular cancer called as leukemia or blood cancer. This cancer tumor iralla. There is no mass of cells. There is just an increase in the number of WBC. So in leukemia, you cannot actually do any biopsy etc ಮಾಡಬೇಕಾಗಿಲ್ಲ. Yes, you can take from the bone marrow etc, but otherwise to diagnose it you don't have to do this. What is done in this case, all that has to be done is you just have to take the blood of the person and put it under a slide. Smear ಅಂತ ಹೇಳ್ತೀವಿ, ಒಂದು ಡ್ರಾಪ್ ಬ್ಲಡ್ ಹಾಕ್ಬಿಟ್ಟು ಈ ತರ ಸ್ಮಿಯರ್ ಮಾಡ್ತೀವಿ. ಮಾಡ್ಬಿಟ್ಟು you will check it under the microscope and count the number of WBCs. When you count it per mm cube, immediately you will come to know whether it is abnormally high. ಸ್ವಲ್ಪ high ಇದ್ರೆ maybe because of infection. ತುಂಬಾ high ಇದ್ರೆ you will know, and plus they will look different. So based on that, by a blood smear you will be able to identify leukemia or blood cancer. The third method is going to be through X-ray, right? So X-radiations. So what has to be done? Let us say, Vaga, I was telling you that a person, they've been coughing a lot and lot of problems is there. Blood is there in the cough, sputum, etc. So somebody thinks maybe they have lung cancer. Avagain madbeku, a person now ನಿಲ್ಲಿಸ್ಬಿಟ್ಟು, from one side you send X-rays into his body, and they will all appear on a film, and our film na develop mad takshana you will come to know whether it looks like a normal lung or whether there is any mass. Okay, abnormal yavdaru mass kanastu andre immediately you will come to know that there is cancer. X-rays, the students may know that in X-rays what is basically going to happen is you find that X-rays, certain solid substances like bones etc are going to be opaque to it, and air, lungs are filled with air, no? So what happens is X-rays neatly go through the lungs, hangagi all those parts appear dark in color. So hagage a person who has got trained in X-ray technology will be able to identify whether this is a mass illa sumne fluid fill agbitidya, like as in pneumonia etc. They will be able to identify. So that is going to be X-ray, right? Next, fourth one is called as CT scan, computed tomography scan. ಇಲ್ಲ ಕಂಪ್ಯೂಟೆಡ್ ಆಕ್ಸಿಯಲ್ ಟೊಮೊಗ್ರಫಿ ಅಂತಾನು ಅನ್ಬೋದು, or CT scan ಅಂತ ಹೇಳ್ತೀವಿ. What is CT scan? CT scan works on the same principle as X-ray. There is no other difference between them. Okay, but what CT scan is able to do is like this. Suppose ಈಗ ನನಗೆ I am suspected with lung cancer. What if a tumor is hiding behind the ribs? Other location of the tumor is such that it is hiding behind the ribs. When I take an X-ray, I'm not able to make out at all. So in such a case, what can be done is you can send X-ray to 360 degree of the body. ಓಕೆ, ಒಂದು CT ಸ್ಕ್ಯಾನ್ ಮಷೀನ್ ಅಂತ ಇರುತ್ತೆ. ಅದರಲ್ಲಿ ಪರ್ಸನ್ ನ ಮಲಗಿಸ್ಬಿಟ್ಟು 360 ಡಿಗ್ರಿಯಿಂದ ವಿ ಸೆಂಡ್ ದ ಎಕ್ಸ್ರೇ. ಸೊ ಇಲ್ಲಿಂದಾನು ಎಕ್ಸ್ರೇ ಕಳ್ಸೋದು, ಇಲ್ಲಿಂದಾನು ಕಳ್ಸೋದು, ಇಲ್ಲಿಂದಾನು ಕಳ್ಸೋದು. ಸೊ ಥ್ರೂ ಆಲ್ ದೀಸ್ ಲೊಕೇಶನ್ಸ್ ವೆನ್ ದಿ ಎಕ್ಸ್ರೇ ಇಸ್ ಸೆಂಟ್ ಅಂಡ್ ಆನ್ ದಿ ಆಪೋಸಿಟ್ ಸೈಡ್, ಇಲ್ಲಿಂದ ಎಕ್ಸ್ರೇ ಬಂದ್ರೆ ಇಲ್ಲಿ ಆಪೋಸಿಟ್ ಸೈಡ್ ಅಲ್ಲಿ ದೇರ್ ವಿಲ್ ಬಿ ದ ಡಿಟೆಕ್ಟರ್. ಯೂಸಿಂಗ್ ದಟ್ computed. So using a software, that information is compiled into an image. So it is like you get slices of the body, neatagi slices of the body thara image barutte. That is going to be called as computed tomography. Avaga enagutte andre smallest tumor neatagi detect agbidabodu. But please understand, CT scan is very affordable. It is very tumba bega madbidabodu. Within a few minutes only CT scan madbodu. But if you want to check whether the disease tumor kadme aagta idya antella check madake CT scan use madalla, because by itself it is going to be having what do we say, x-rays irodrinda it might become dangerous. So hangagi once it can be done, that is not going to be an issue. The next diagnosis is going to be called as MRI, magnetic resonance imaging anta. Okay, KSTL I had given a lecture on imaging techniques. So some of the students, those who are interested, you can go through that, and adrandinu nimge you will get to know what this is. Cancer ali you just need to know the names. MRI, magnetic resonance imaging, andre the body is subjected to a highly electromagnetic field. So hangagi enaagbidutte andre you find that the protons in the body get aligned. When they get realigned, the extra energy comes out, and we can grossly call it as a resonance that comes out anta helbodu. It is basically radio waves which come out. Adanna detect ಮಾಡಿ you can form the image of the body. ಬಹಳ ಸೇಫು MRI. You can use it even for a pregnant woman. ಚಿಕ್ಕ ಮಕ್ಕಳಿಗೂ ಯೂಸ್ ಮಾಡಬಹುದು. Only thing is it takes a long time. ಬಹಳ ಟೈಮ್ ತಗೊಳುತ್ತೆ MRI ಮಾಡಕ್ಕೆ. 20 minutes ತಗೋಬಹುದು, 15-20 minutes ತಗೋಬಹುದು. That is one thing. Any person who has a pacemaker etc ಅವ್ರಿಗೆ ಮಾಡಕ್ಕಾಗಲ್ಲ. That is MRI. Images are very clear. Okay, so like this is going to be the next one. Last diagnosis is going to be through antibodies or monoclonal antibodies anta heltivi, which are they are cancer specific. Okay, and they are made radio labeled. So our radio labeled antibodies, now if we send them into the body or on a group of cells, let us say there is a tumor, it looks like normal but it is a tumor, but I want to know if it is cancerous. So radio labeled antibodies based on the cancer, if I add, and if it binds and there is radioactivity, immediately we will come to know that there is cancer. Okay, so this is how cancer can be diagnosed, right? So I think ಈಗ almost 4:50 ಆಗೋಗ್ತಾ ಇದೆ. Let me just put on the ನಿಮ್ಗೋಸ್ಕರ ಒಂದು ಇದನ್ನ ಮಾಡಿದ್ದೀನಿ presentation. We will look at some images. After that I will discuss some of the questions that can be asked for NEET exam ಇರ್ಬೋದು, KCET exam ಇರ್ಬೋದು. ಆ ತರದ್ದು questions ಒಂದೊಂದನ್ನ ನಿಮ್ಗೆ ಕೇಳ್ತೀನಿ. Okay, right?
ಹೇಳಿ ಮೇಡಮ್.
ಇಲ್ಲಿ ಶೇರ್ ಮಾಡ್ತೀನಿ ಸರ್, ಒಂದ್ ನಿಮಿಷ. ಐ ಜಸ್ಟ್ ಶೇರ್ ಸ್ಕ್ರೀನ್ ನ ಶೇರ್ ಮಾಡ್ತೀನಿ. ಎಸ್ ಮಕ್ಕಳೇ, ಐ ಥಿಂಕ್ ಯು ಆರ್ ಏಬಲ್ ಟು ಸಿ ಆನ್ ಯೋರ್ ಸ್ಕ್ರೀನ್. ನೀವು ನೋಡ್ಬೋದು ಆನ್ ದ ಸ್ಕ್ರೀನ್. ನೋಡಕ್ ಆಗ್ತಾ ಇದೆಯಾ ಸರ್? ನಿಮಗ್ ನೋಡಕ್ ಆಗ್ತಾ ಇದೆಯಾ ನಂದು ಸ್ಕ್ರೀನು?
ಫುಲ್ ಸ್ಕ್ರೀನ್ ಮಾಡ್ಕೊಳ್ಳಿ ಮೇಡಮ್.
ಹಾ, ಕಾಣಿಸ್ತಾ ಇದೆಯಾ? I have shared the presentation. Are you able to see? ನನಗೆ sharing ಅಂತ ಬಂದಿದೆ. You are able to see, no? Yes, thank you so much. Okay, so cancer ದು ನೋಡೋಣ ಒಂದ್ಸತಿ. what are the things? ಇಲ್ಲಿ ನೋಡಿ children, you can see on your left hand side how normal cells look like, whereas cancerous cells ಅದರದ್ದು shape ಬೇರೆ ಇರುತ್ತೆ, structure ಬೇರೆ ಇರುತ್ತೆ, and even the size will be very different. Yes sir?
ಏನಿಲ್ಲ ಮ್ಯಾಮ್, ಕಂಟಿನ್ಯೂ ಪ್ಲೀಸ್.
Yes, yes, thank you. So cancerous cells will look very different. So adralle train agirtare. There are diagnostic labs ali, there are technicians and they will be trained to identify, right? This is an image where you can see the yellow cell in the middle is a big cancerous cell and all the small cells which are present outside, they are all going to be your WBC. They are trying to attack the cancerous cell and they are trying to destroy it, right? So remember, every person may have one cancerous cell produce agbodu, but ellargu cancer barde irbodu. That is mainly because of good immunity. Immune cells are able to detect, but if it goes out of control, avaga disease called cancer agate, right? Here you can see when the petri dish togondre, normal cells idre, they will just divide to make a layer cover up, they will not divide. Whereas cancerous cells, layer cover admeluve, they will continue to divide to make like a mountain. So cancerous cells ge contact inhibition iralla. Benign tumor andre eno? It is present in a particular location. It will not spread at all. Cancer no word we cannot use for a benign tumor unless it is present in a very critical part. Then we have to treat. Malignant tumor andre, the cells will come away and they will spread to other parts, and that is called metastasis, which is a, as I said, it's a feared property, right? Histopathology andre, you can see on the screen, left hand side top ali, one small part of the liver can be taken. Amele you will put it in a wax. Wax ಅಲ್ಲಿ ಹಾಕಿದ ತಕ್ಷಣ ಅದನ್ನ wax impregnation ಅಂತ ಅಂತೀವಿ. Then you can see at the bottom this person is taking slice ತಗೊಂಡು then we will study it under the microscope. ಅದು histopathology ಅಂತ ಅಂತೀವಿ. ಇದು blood smear, okay? Normal blood left hand side ಅಲ್ಲಿ ಕಾಣಿಸುತ್ತೆ ನಿಮ್ಗೆ, where you find that the number of blood white WBC are very less. A number of RBC will be more. Whereas cancerous ali, you can see the cells look very different also. ಬಹಳ ಡಿಫರೆಂಟ್ ಆಗಿ ಕಾಣಿಸ್ತಿದೆ and the number of cells are huge. Okay, that is going to be blood smear we do to detect leukemia, right? X-ray nu ಮಾಡಬಹುದು. So on the left hand side you can see the person is standing, a chest X-ray is being taken, right? And we generate X-rays in the machine and then we send it through the person. ಈ ಕಡೆ I have taken this image. It is a large white, it's a very aggressive tumor that is growing. Nimge nodakagato ilvo gottilla. Left hand side lung ali, there is a very big tumor which is present. Left lung ali, you have a very big tumor that is present there. It is a big, large tumor that is present. White giant anta antare. Okay, this is how a CT scan is going to be. CT scan machine, right hand side alli nodbodu. A person na kalsadre X-ray source will rotate, detector will rotate, and we will be able to see the images clearly. Okay, ade tara MRI barutte. The machine looks the same, but the principle is totally different. Idralli electromagnetic waves generate agate, and because of that, you can, once they pass through the body, na heldange, the energy is given out when the protons will realign and you can detect it as radio waves. Adu MRI. ಈ ತರ antibodies ಇರುತ್ತೆ. We call them as monoclonal antibody ಅಂತ ಹೇಳ್ತೀವಿ, which are very specific to cancer proteins. So ಅದನ್ನ ಹಾಕಿದ್ರೆ immediately they will be able to detect. Okay, right? I'll just stop sharing. Right. Okay, sir. I think I'm done. I will go to some questions for the students. Students question ಕೇಳಿಸ್ಕೊಳ್ಳಿ. And if you want, you can even write down the questions, not a problem. What are the type of questions that can come? First board exam ಮಾಡ್ಬಿಡೋಣ. Easy ಅಲ್ವಾ board exam? ಅದರದ್ದು ಮಾಡೋಣ. So board exam questions that can be asked is, they can ask you difference between benign tumor and malignant tumor. Question number 1. What is the difference between benign tumor and malignant tumor? That can be one question which can be asked. Okay. Second question that can be asked with respect to cancer is, list any 4 methods of detecting or diagnosing cancer. List any 4 methods of detecting or diagnosing cancer. This also can be asked in the board exam, right? Innondu question that can come in board exam is going to be, what is metastasis? What is metastasis? This is one more question that can come in the board exam. Now let us go to KCET type of question. KCET type of question yav thara keltare? They may ask question like, what is the full form of MRI? MRI the full form magnetic resonance imaging. ಈ ತರ simple questions KCET li ಬರುತ್ತೆ. What is the full form of MRI? ಆ ತರ ಬರ್ಬೋದು KCET li. ಆಮೇಲೆ next KCET li ಇನ್ನೊಂದು question ಯಾವ್ದನ್ನ definitely they may ask is, they may ask what is biopsy? What is biopsy? ಅಂತ ಕೇಳಿ choices ಕೊಡ್ಬೋದು. So choices ಏನ್ ಕೊಡ್ಬೋದು? Choices can be treatment of cancer is biopsy. Formation of cancer is biopsy. Removal of a tissue from the body to diagnose cancer is biopsy. Okay, and last can be treatment of cancer using radiations is called as biopsy. Ee thara some choices may be given, right? It is a very simple type of question. Neatly solpa high level questions kelbodu. Yav tara kelbodu? There is a person there is a person who got exposed to certain chemicals that are carcinogens. Which of these cancers is most likely to be formed anta kelbodu? Okay, one thing you have to understand, students, 70% of the cancers come from epithelial tissues and we call them as carcinomas. Okay, the remaining type of cancers can be from the connective tissue irbodu. Yes, neural tissue irbodu. Those will be the remaining 30% of cancers. 70% of cancers are going to be epithelial tissue cancers. Yake? Neatly they may say, why epithelial tissue cancers are more common than cancers of connective tissue or cancers of muscle tissue or cancers of neural tissue anta question barbodu. So the question, the answer is going to be, you have to apply first year concept of mitosis and second year concept of cancer. So the reason is epithelial tissues by themselves show high rate of cell division. So in case they go out of hand, WBC will not be able to detect. ಯಾವ ತರ ಅಂದ್ರೆ ದಿನಾನು WBC is seeing epithelial tissue cell is dividing. So it will ignore if it divides a little bit more. Okay, whereas if it is, let us say, a cancer of the bone cancer bandre, immediately WBC will realize there is something wrong because bone cells don't divide all the time. So like this, a question can come in NEET. Another question that can come in NEET is, they can ask you, what is the difference between C-onc and V-onc? As a multiple choice question they can ask you. So C-onc is cellular oncogene formed by the mutation of proto-oncogene. Correct answer. Or they can say C-onc gene is present in radiations. Completely nonsense answer. Or they may say C-onc gene is present in normal cells. Also a wrong answer. They may say V-onc genes are seen because they are mainly present in vultures. Again a wrong answer. Like this, some question can be asked. Another question that can come in NEET exam is, they can definitely ask you regarding CT scan. Okay, so they may say CT scan is used for what? Okay, MRI full form, CT scan full form idella CET il barbodu, but NEET exam alli CT scan is used for what anta kodbodu. So you may find that, yes, can I use CT scan even to find out whether there is bleeding in the brain? Khandita. But NEET exam alli they expect the answer to be there in NCERT. NCERT li bleeding in the brain ಬಗ್ಗೆ mention ಆಗಿಲ್ಲ, as well as not CT scan used for that anta mention agilla. So like this, some questions can be asked. Okay, yes sir, over to you.
Okay, thank you, ma'am.
What are the primary symptoms of cancer? Yogesh, very good question.
Yes.
Primary symptoms of cancer tumba important, Yogesh, and ivaga MBBS colleges alli they are trying to spread the awareness to the local community. Yogesh, it depends on what type of cancer. Suppose skin cancer ittu andre, there will be a black mass formed which will continue to grow very fast. Correct? There will be a mass form which will continue to grow very fast. That can be an indication of skin cancer. Lung cancer adre enagate, the person will feel very tired if they climb the steps because our respiratory exchange is getting affected. But That can be seen even if a person has TB. That can be seen even if a person has pneumonia. ಅದಕ್ಕೆ ಡಯಾಗ್ನೋಸ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಓಕೆ, ದಟ್ ಇಸ್ ಒನ್ ಥಿಂಗ್. ಸೊ ಲಂಗ್ ಕ್ಯಾನ್ಸರ್ ಅಲ್ಲಿ ಮೇ ಬಿ ದೇರ್ ವಿಲ್ ಬಿ ಅ ಲಾಟ್ ಆಫ್ ಬ್ಲಡ್ ದಟ್ ಕ್ಯಾನ್ ಕಮ್ ಇನ್ ದ ಸ್ಪೂಟಮ್. ದಟ್ ಮೇ ಬಿ ದೇರ್, ಬಟ್ ಆ ಸ್ಟೇಜ್ ರೀಚ್ ಆಗಕ್ಕೆ ಯಾರು ಬಿಡಲ್ಲ. ದೇ ಮೇ ಗೆಟ್ ಇಟ್ ಡಯಾಗ್ನೋಸ್ಡ್. ಇಫ್ ಇಟ್ ಇಸ್ ಓರಲ್ ಕ್ಯಾನ್ಸರ್, ಓರಲ್ ಕ್ಯಾನ್ಸರ್ ಆದ್ರೆ ಏನಾಗುತ್ತೆ, ಒಳಗಡೆ ದಪ್ಪ ದಪ್ಪ ಮಾಸ್ ಇರುತ್ತೆ. ನೀವು ನೋಡಿರ್ಬೋದು, ಸಿಗರೆಟ್ ಸ್ಮೋಕಿಂಗ್ ಮಾಡಬಾರ್ದು ಅಂತ ದೇ ಶೋ ದೋಸ್ ಆಡ್ಸ್ ನೋ ವಿತ್ ವೆರಿ ಬಿಗ್ ಮಾಸಸ್. ಆ ತರ ಇರ್ಬೋದು. ಥ್ರೋಟ್ ಕ್ಯಾನ್ಸರ್ ಆದ್ರೆ ದ ಪರ್ಸನ್ ವಿಲ್ ನಾಟ್ ಬಿ ಏಬಲ್ ಟು ಸ್ವಾಲೋ ಎಟ್ ಆಲ್ ಆರ್ ಸ್ಪೀಕ್ ಪ್ರಾಪರ್ಲಿ. ಕರೆಕ್ಟ್, ಅದ ಆಗ್ಬೋದು. ಬ್ಲಡ್ ಕ್ಯಾನ್ಸರ್ ಆದ್ರೆ ದ ಪರ್ಸನ್ ವಿಲ್ ಬಿ ಸೋ ಟಯರ್ಡ್, ಅನ್ಬಿಲೀವಬ್ಲಿ ಟಯರ್ಡ್, ಬಿಕಾಸ್ RBC ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇರಲ್ಲ. ಸೊ ಎವ್ರಿ ಕ್ಯಾನ್ಸರ್ಗೆ ಸಿಂಪ್ಟಮ್ಸ್ ಬೇರೆ ಬೇರೆ ಬರ್ಬೋದು. ಓಕೆ, ಇಸ್ ದೇರ್ ಅನ್ ಇನ್ಕ್ರೀಸ್ ಇನ್ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ ಇನ್ ಕ್ಯಾನ್ಸರ್? ಇಲ್ಲಮ್ಮ, ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ ಇನ್ಕ್ರೀಸ್ ಆಗಲ್ಲ. ಬ್ಲಡ್ ಕ್ಯಾನ್ಸರ್ ಅಲ್ಲಿ ಎಬ್ನಾರ್ಮಲ್ ಕ್ರೋಮೋಸೋಮ್ ಮೇ ಕಮ್. ಓಕೆ, ಫಿಲಡೆಲ್ಫಿಯಾ ಕ್ರೋಮೋಸೋಮ್ ಅಂತ ಹೇಳ್ತೀವಿ, ವಿಚ್ ಇಸ್ ಸೀನ್ ಇನ್ ಸರ್ಟನ್ ಟೈಪ್ ಆಫ್ ಲುಕೇಮಿಯಾ. ಎಲ್ಲ ಲುಕೇಮಿಯಾ ಅಲ್ಲ, ಸರ್ಟನ್ ಟೈಪ್ ಆಫ್ ಲುಕೇಮಿಯಾ ಯು ಫೈಂಡ್ ದಟ್ ದೇರ್ ವಿಲ್ ಬಿ ಫಿಲಡೆಲ್ಫಿಯಾ ಕ್ರೋಮೋಸೋಮ್ ಅಂತ ಇರ್ಬೋದು. ಐ ಥಿಂಕ್ ಸಮ್ ಮೋರ್ ಕ್ವೆಶನ್ಸ್ ಆರ್ ದೇರ್. ಇಲ್ಲಿ ಹೈಡ್ ಆಗಿದೆ, ಐ ಜಸ್ಟ್ ಚೆಕ್. ಬ್ಲಡ್ ಸ್ಮಿಯರ್ ಅಂದ್ರೆ ಏನು ಅಂದ್ರೆ, ಬ್ಲಡ್ ನ ಈಗ ಅದ್ಕೊಳ್ಳಿ, ಇದು ನಂದು ಫೋನ್ ಇದೆ, ಇದು ಒಂದು ಸ್ಲೈಡು, ಇದ್ರ ಮೇಲೆ ಒಂದು ಡ್ರಾಪ್ ಆಫ್ ಬ್ಲಡ್ ಹಾಕ್ತಿವಿ, ಇದು ಇನ್ನೊಂದು ಸ್ಲೈಡು, ಹಿಂಗೆ ಸ್ಮಿಯರ್ ಮಾಡ್ತೀವಿ. ಸ್ಮಿಯರ್ ಮಾಡಿದ್ರೆ ಥಿನ್ ಫಿಲ್ಮ್ ಆಫ್ ಬ್ಲಡ್ ಬರುತ್ತೆ. ದೆನ್ ಯು ಹ್ಯಾವ್ ಟು ಕೌಂಟ್ ದ ವಾಟ್ ಯು ಸೇ ದ ಬ್ಲಡ್ ಸೆಲ್ಸ್. ಅದಕ್ಕೆ ನಾರ್ಮಲ್ ಬ್ಲಡ್ ಸ್ಲೈಡ್ ತಗೊಳಕ್ ಆಗಲ್ಲ ಶಿವರಾಜ್. ಯು ಹ್ಯಾವ್ ಟು ಟೇಕ್ ಒಂದು ಸ್ಪೆಷಲ್ ಟೈಪ್ ಆಫ್ ಸ್ಲೈಡ್ ಸಿಗುತ್ತೆ ವಿಚ್ ಹ್ಯಾಸ್ ಗಾಟ್ ಗ್ರಾಫ್ ಲೈನ್ಸ್ ಆನ್ ಇಟ್. ಆ ಸ್ಲೈಡ್ ನ ತಗೊಂಡ್ರೆ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಕೌಂಟ್. ಅದಕ್ಕೆ ಬ್ಲಡ್ ಸ್ಮಿಯರ್ ಅಂತ ಅಂತೀವಿ. ಓಕೆ, ಇನ್ನೊಂದು ಕ್ವೆಶ್ಚನ್ ಇದ್ದಿದ್ದು, ಇಸ್ ದೇರ್ ಇನ್ಕ್ರೀಸ್ ಇನ್ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್? ಹೇಳಿದ್ದೀನಿ. ಆರ್ ಆಲ್ ಕ್ಯಾನ್ಸರ್ಸ್ ಕ್ಯೂರಬಲ್? ಥಿಯರಾಟಿಕಲಿ ಎಸ್ ಜ್ಯೋತಿ, ಆಲ್ ಕ್ಯಾನ್ಸರ್ಸ್ ಥಿಯರಾಟಿಕಲಿ ಆರ್ ಕ್ಯೂರಬಲ್. ಬಟ್ ಏನಾಗ್ಬಿಡುತ್ತೆ ಅಂದ್ರೆ, it is a fight between the cancerous cells and the treatment, correct? So sometimes it happens that we are not able to reach the cancer cells with respect to treatment. Okay, ಇವತ್ತಿನ ದಿನ ನಾನು ಟ್ರೀಟ್ಮೆಂಟ್ ಬಗ್ಗೆ ಮಾತೇ ಆಡಿಲ್ಲ, because I knew that it will take another half an hour to 45 minutes. Maybe ಇನ್ನೊಂದ್ಸತಿ ಚಾನ್ಸ್ ಸಿಕ್ಕರೆ I will be able to speak about cancer treatment also. Right, so all are curable andre theoretically yes, but sadly many people still die from cancer. Okay, awareness kadme irbodu, so lot of reasons may be there. Okay, what are the primary symptoms helidini? Okay, I think that's it. I think yes, thank you so much. Thank you for the questions also, students. It was a pleasure interacting with you. Yes sir.
Okay, thank you ma'am.
Thank you so much sir.
ಸೊ ಇವತ್ತಿನ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ನೀವು ಕ್ಯಾನ್ಸರ್ ಅಂದ್ರೆ ಏನು, ಕ್ಯಾನ್ಸರ್ ಯಾವ ರೀತಿ ಬರುತ್ತೆ, ಮತ್ತೆ ಅದನ್ನ ಯಾವ ರೀತಿ ನಾವು ಕ್ಯೂರ್
ಡಿಟೆಕ್ಟ್ ಮಾಡಬಹುದು,
ಎಸ್ ಮತ್ತೆ ಡಿಟೆಕ್ಟ್ ಮಾಡಬಹುದು ಅನ್ನೋ ಒಂದು ಹಲವಾರು ವಿಚಾರಗಳನ್ನ ಮತ್ತು ಕನ್ನಡ ಇಂಗ್ಲಿಷ್ ಎರಡರಲ್ಲೂ ಕೂಡ ಭಾರಿ ಸರಳವಾಗಿ ಮಕ್ಕಳಿಗೆ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ರಿ ಮೇಡಮ್. ಸೊ ಇಟ್ಸ್ ವೆರಿ ಇನ್ಫಾರ್ಮೇಟಿವ್ ಟಾಕ್ಸ್ ಆನ್ ಕ್ಯಾನ್ಸರ್. ಸೊ ಬಿಹಾಫ್ ಆಫ್ KST and myself, thank you ma'am.
Thank you so much sir, thank you so much. All the best dear students, all the best. Thank you sir, thank you.
ಇವತ್ತಿನ ಹಾಜರಾತಿಯಲ್ಲಿ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳು ಕೂಡ ಹಲವಾರು ಕ್ವಶನ್ಸ್ ಗಳನ್ನ ಪ್ರಶ್ನೆಗಳನ್ನ ಕೇಳಿದ್ರೆ, ಇದೆ ಫಸ್ಟ್ ಟೈಮ್ ಇಷ್ಟೊಂದು ಕ್ವಶನ್ಸ್ ಕೇಳಿರೋದು ಮ್ಯಾಮ್, ನಂಗೆ ಭಾರಿ ಸಂತೋಷ ಆಯ್ತು.
Yes sir, yes sir, yes sir.
ಸೊ ಇದೇ ರೀತಿ ಎಲ್ಲಾ ವಿಷಯಗಳಲ್ಲೂ ಕೂಡ ಕ್ವಶನ್ಸ್ ಕೇಳಬೇಕಂತ ಮತ್ತೊಮ್ಮೆ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ತಿಳಿಸುತ್ತಾ, ಸೊ ನಾಳೆ ಯಾವುದೇ ವರಮಹಾಲಕ್ಷ್ಮಿ ಹಬ್ಬದ ಪ್ರಯುಕ್ತ ಎಲ್ಲರಿಗೂ ರಜೆ ಇರುತ್ತೆ ಈ ಕ್ಲಾಸ್ ಪ್ರಯುಕ್ತ. ಸೊ ಥ್ಯಾಂಕ್ ಯು ಸ್ಟೂಡೆಂಟ್ಸ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್, ಥ್ಯಾಂಕ್ ಯು. ತಮಗೂ ಹಬ್ಬದ ಶುಭಾಶಯಗಳು. ಥ್ಯಾಂಕ್ ಯು
ಸೇಮ್ ಟು ಯು ಮ್ಯಾಮ್. ಸೊ ಈ ಕಾರ್ಯಕ್ರಮದ ರೂವಾರಿಗಳಾದಂತಹ ನಮ್ಮ CEO ಆದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಸೊ ಥ್ಯಾಂಕ್ ಯು ಒನ್ಸ್ ಅಗೇನ್. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್.